Okay, thank you, Nick. Thank you, everybody. Good afternoon, if you're in the same time zone as those of us in the room. Target product profiles for conventional antivenoms. So I put this slide in, and it's completely redundant because David's already presented this. This was simply to posit the work in the broader context of what WHO is trying to do these days. This is the real starting point for this. Now, some of you may disagree with that rather slightly dramatic title. I think, unfortunately, regrettably, sadly, it is a defensible title. But the important thing is that that circle on the left is not a circle, it's a spiral, and it's currently been going down for a number of years. And our role collectively as a group of individuals and as a group of scientists and clinicians is to reverse that, so it's a spiral that's going back up again. I think we would all agree that as I've tried to very politely put it, there's a variable quality of antivenoms out there on the market. The good ones are very, very good indeed, and I've yet to meet a clinician who would not wish to have one when they've got a patient in front of them. And let's face it, get clinicians to agree on so little things these days that getting them to agree on what suggests it may even be true. But then we try and look at what are the underlying reasons why some other antivenoms are considered very poor. And the reason is because, paradoxically, because they're actually a very old product, many of them in some ways predate modern regulatory guidance and oversight. And it's one of these sort of historical quirks that if you have a product on the market and regulations change, you don't actually necessarily have to retrospectively change it, depending on your jurisdiction, but that's a general rule. So one way we can try to improve things is to provide some guidance on what a conventional antivenom really should look like. And that's where target product profiles come in. Those of you with an industrial background will be starting to sort of mentally yawn at this point because they're utterly ubiquitous. They are a core part of what you would always do. Those of us from a more academic background, these things also exist. There are different flavors of types of target product profiles, or TPPs. And what we're working with in this context is a public sector TPP. Now, they're usually put in place before you start an R&D process to try and find a new product, whether that be a pharmaceutical, medical product, or a diagnostic. But they can also be used for existing products, and that's the context of this talk. And because this is a broad audience, I thought it'd be a good idea to add in at least one definition of what a TPP actually is. It's the desired profile of characteristics of a target product. That does not mean, if you're from my sort of background, meaning the biochemical profile, it's much, much broader. It's who are the patients these are intended for? What is the market? What should the packaging look like? What should the biochemistry look like? It's the full package. Before I actually introduce the TPP we've been developing, just two slides on the way we've gone about it. The first is a landscape review, which is put together by the Secretariat. The Secretariat in this case is staff at WHO, David Williams, myself, Raphael, were deeply involved in this, together with two colleagues from Drugs for Neglected Diseases Initiative, DNDI, Isabella Ribeiro and Belén Pedric. We then put together a technical and science advisory group with very wide expertise to try and guide us and stop us making the worst of the possible mistakes we might have, and then draft a TPP. And that is all done by the Secretariat and the TSAG working collectively. Then goes out to public consultation and then eventually publication. And we dearly hope that the first one will be actually out in the public domain next month. Well, we're hoping. When we hope that last month as well, to be fair. But anyway, we're very close, right? This is the Technical Science Advisory Group. If you recognize all the names on that list, we have clearly failed, because the whole point is it's a very broad spectrum of people with lots of different wonderfully interesting backgrounds. Yes, there are some people with deep snakebite expertise, and there are people here who do not have snakebite expertise, and that's deliberate. It has to be broad. Otherwise, you open up your risk to failure. And then you have to decide where to start. And what we decided, we would start with a regional approach. And the data on the right, it happens to be one very decent source. The other sources still give you the same general picture. If in doubt, you start with either sub-Saharan Africa or Asia, meaning South Asia or Southeast Asia. We chose to work with sub-Saharan Africa first, and South Asia is coming next. We also chose to focus on a more limited array of species rather than trying to do every venomous snake out there. There has been in place for a number of years now a WHO classification, and we chose to work on the category 1 and 2 snakes only for each region. In sub-Saharan Africa, that's 24 in each category. That's 48 species in total. It provides a starting point and some measure of focus. And this is what we've eventually come up with. We've not come up with a TPP for sub-Saharan Africa. We've come up with 4. Now, the first 2, I suspect, are very familiar to an audience such as this. It's a broad-spectrum Pan-African polyvalent. So irrespective of what the species of snake is that may have bitten the poor patient who's arrived in front of a clinician, this is what you might go for first. And as we all know, there are some cases where a monovalent product can also have considerable utility if you happen to know what that species is, especially if it's a species that's not well covered by a polyvalent product. And then we have chosen to add in 2 more categories. And this illustrates where a TPP is not only telling us where we are now, but trying to give some measure of direction of where we might want to go in the future. There are no products in either of these 2 categories in sub-Saharan Africa, but in other parts of the world, it has been an approach that's proved very effective, which is to look at patients who are either largely neurotoxic envenoming or largely non-neurotoxic, meaning procoagulant hemorrhagic cytotoxic. So the fact that no products exist now doesn't matter. The important point is if a manufacturer is interested in going out and making such a product, they have some guidance to help them start on that process. And this slide takes you into what a TPP starts to look like. No, you can't read it. Don't worry, you're not meant to. This is simply the first 4 categories in the TPP document. What I hope you can see, though, is that there are 3 columns. The first tells us what criteria we're working with, what characteristic, and then there's optimal and minimal. The minimal is what we think is completely plausible, acceptable, based on products that are already out there. The optimal is what we think is potentially achievable. So there is a balance between ambition and between practicality in the optimal category. And for some characteristics, there's no point making a distinction, and that's why we haven't bothered. Now, don't panic. There are 21 line items that are common to all of them. Then there's another 4 for each of them. There's 37 line items and 42 pages, and I'm not going to go through all of them. So what I'm going to do is just pick out a couple of things which I think well illustrate points I want to make, because I think they're interesting for TPPs. And the first of these is the active pharmaceutical ingredient. And I picked this one out because the API, in this context, what it ought to be formally is that fraction of the immunoglobulin that binds a venom fraction and has an activity that it neutralizes. But that very specific, correct definition is almost impossible to measure properly, accurately, because, as we all know, venoms are very complex, which means the conventionally derived antisera are also very complex. So what we've done instead is we've used a proxy, an API here, which is unusual compared with TPPs for lots of other diseases you may have come across, which is it's the whole IgG or the Fab2 fragment that is raised. So it's all of it, the whole, as opposed to that which can be shown to specifically bind to venom components and only those. For the minimal, we're happy with either Fab2 fragments, many products are Fab2, or whole IgG, as some are. But we think there is on balance, sorry, we in this context is the secretary, guided by the Technical Science Advisory Group, think that whole IgG is probably the better way to go for the future. The finished product form in this context is either a liquid product or a lyophilized product. Both exist on the market at the moment, as we know. There are advantages to both. We recognize those, and the text will illustrate that. But optimally, we think the lyophilized is probably the better overall direction. And it boils down to it doesn't need a cold chain. And in so many parts of the world where we want these products used, that is a big issue. Not just presence of a cold chain, presence of a reliable cold chain. Immunoglobulin content and protein content. The IG content, we've said 85%, and we said 90%. But the key thing is that the only thing that counts is whole IG or Fab2. If there's a Fab fragments in there, if there's other things in there, and I've seen some data which suggests there are other things in there, like FC fragments or things that might reasonably expect to come out of a purification process that hasn't gone quite as I would have liked it to go. That no longer counts. That's a straightforward guidance for a fairly straightforward assay for a manufacturer. Protein content in there, I put in there simply because it illustrates where there's a distinction to be made. What we've put on the slide applies for the polyvalence, and it applies for if it's either neurotoxic envenoming or non-neurotoxic envenoming, but not for the monovalence, because it has to be a compromise. And we've said 5% or 7.5%. But for the monovalence, you can, and it is sensible to get a little bit clever, because the overall values have to be overall values because you do not know what species of snake was involved. And we know that because that's how we defined it in the first place. And we all know that yields from snakes vary enormously. But with the monovalence, because you know what species you're working with, you've got a much more accurate understanding of what the venom yields might well be, and you can tailor it, which means you can have the conversation with a regulatory agency along that basis. And clinical effectiveness, I've brought in partly because it was an issue that required a great deal of conversation to get to these numbers. As a non-clinician, for me, the important thing was not that we got the clinicians to agree, but the important thing is the reasons why it was difficult. And the real reason is in red at the bottom. This is a field where there have been so few clinical trials, and that's the core problem. What we need is a great deal more clinical trials. What we have got is some actually pretty decent observational reports out there. Some are less good, but there are some good observational studies out there. And that's not enough. In almost any other field that I've worked in, you would default to look at the clinical trial data first, and here it's very difficult to do that. So this is the classic plea for please more clinical trials. And I think we're going to hear something more about at least one of those later in the meeting, which is great. And if the clinical trial data is poor, cost effectiveness, cost benefit analysis is equally poor, possibly even worse. And that's a pretty depressing thing to say. So we have deliberately not said this is what the unit price we think should be. We can't, because the number of units you need for effective treatment varies around the world, varies with the products, varies with the snakes, but mainly it varies with enormously on what the other costs are, and those will vary enormously around the world. So what we've explicitly stated, we're looking for a fair pricing concept, one where manufacturers can make some sort of profit margin because they have to, because otherwise they ain't going to exist. And procurement agencies can buy things at a price point which is reasonable for their government in their context, or whoever is paying for this to pay for, whether it's government or insurance company or whatever. And ultimately, many instances, of course, it is the victim and their families and their neighbors who are paying for this. So what we really need, again, is real cost effectiveness studies. What you'll get, hopefully next month, is that's actually we've changed the front cover, but that's what it was when I put the slide together for Sub-Saharan Africa. Very soon following that, the work on South Asia is progressing, and we would hope to get you there very soon. And then we will literally walk our way around other regions of the world. Because we've got a stable group of 18 people, and they reflect all regions of the world deliberately, a lot of the issues we've talked about for Southern Africa will not need to be replicated because we know they will apply. And as an aside, we're also in parallel working on TPPs for small molecule therapeutics. and for what I've called engineered antibodies, which effectively is humanized monoclonals, nanobodies, and peptide aptamers. So without any further ado, thank you to the Technical Science Advisor Group. Thank you to Wellcome for funding this. To my colleagues at DNDi, that's Isabella and Belen, and to my colleagues at DHO, that's David, Raphael, Beth, who are in the room, and Bernadette, who is our glorious leader, who can't be with us, unfortunately. Thank you.
Thank you, Mike. Quick question in the room for Mike at the back. Microphone's coming.
You can mention your name and location before you ask the question.
Very much. Hello, I'm Leah and I'm from Oxford Brookes University. Mike, thank you so much for speaking and what an incredible amount of work you and the team have done. This is a very simple question, to be honest with you, but out of interest, what was the determining factor to pick sub-Saharan Africa over Asia? And I ask this mostly because due to the amount of deaths, I thought perhaps Asia would have been the place to start in, but
Yep, no, fair point. I mean, and that was one of the criteria. And the other one was, what is even worse provided with current products than the other region? And people who know more about these things need guide us. However bad it is in South Asia, in Sub-Saharan Africa, it's arguably even worse. How depressing is that?
very.
Yeah. But South Asia will be coming soon. Yeah.
Thank you very much.
Thank you. And let's just go online, see if there's a question. So unmute and ask. First come, first served.
Hi, can you hear me?
Yep, go ahead. Thank you.
Yeah, I'm Siddharth and I work at Venoms Bioproducts. My question to you is that in one of your slides, you've mentioned that the optimal antivenom would be a whole IgG. But the general contention is that whole IgG, the FC region might be responsible for adverse reactions. So did it change throughout the literature currently?
Siddharth, thank you. Very good question. Perfectly understand where you're coming from. Conceptually, I completely understand where you are. If you've got a great FC region at the end of your molecule and it's from a horse, wouldn't a human respond to that? And the answer is yes, there is that potential. I think the best data actually mainly comes from South America, in that, yes, that is an issue, but there is a contrasting issue, which is because Fab2 is a smaller molecule, it has a shorter half-life. And to compensate for that, you have to put more material in. So you end up putting more units of that. So you actually end up putting in as much or more to compensate for the shorter half-life. And that was the rationale why the group went for whole IgG as the ideal, but we accept both as the minimal. So we're not expecting any manufacturer to suddenly turn the ship around and change their manufacturing right now. We're flagging for the future, this may be a better direction. I hope that answers your question, Siddharth.
Yes, I just have a follow-up question here. So would the mixture of Fab2 and whole IgG be better because it will have advantage of both the components?
Okay, it will have the advantages and will have the equal disadvantages. I don't know anyone who's ever actually done that study and done the kinetics. I would like to see the kinetics personally. But there are other, I'm looking around the room to see if other people would agree or disagree with that.
Agree.
Nick would agree, he's ex-industry. He ought to know more about this than me.
And on that one, thank you very much.
Thank you.
Thanks, Mike.